So hello students, welcome to your own channel Vedantu NEET English. This is Baswaraj sir, your biology master teacher. So before we start today's class, let me know quickly right now in the chat if my voice is audible to all of you and the presentation behind me is visible to each and every one of you. This is a session where we do biomolecules, right? Biomolecules is one of those chapters which requires dedication and understanding the concepts. You simply cannot mug up this chapter, and henceforth students believe that this chapter is a difficult chapter. But let me prove you wrong. After today's class, you will be like, sir, this is such an easy chapter. The NCERT is so easy to read, right? NCERT is so easy to read after today's session. Students, quickly go and grab your NCERT, grab a pen in your hand, grab a water bottle and sit down for an amazing class today, right? Amazing class today. Hello, students. Hello. Let me know in the chat if my voice is audible to all of you and the presentation behind me is visible to all of you. Let me know in the chat quickly. Let me also check on my own. Sometimes scam can happen. Right? Let me know if I'm audible and visible also. Yes, I am audible and visible to all of you also. Yes, I'm audible and visible. And also happy Ganesh Chaturthi everyone. Happy Ganesha Chaturthi for each and every one of you. Right? So with that being said, students, can we start today's class? Can we start today's amazing class? Can you let me know in the chat? Hello. Yes. Good morning. Happy Ganesh Chaturthi to each and every one of you. May the Lord Ganesha bless all of you. Right. Now, students, I will be reading the lines of NCERT. So all of you should read with me. That is a simple thing with how this class is going to go. Right. When I read a line of NCERT, I want you also to read the line of NCERT with me. So you'll understand better and you will not cheat yourself. Right. Nice. Now this is a class obviously 11th, right? Obviously class 11th. Now, students, can anyone tell me what is the meaning of biomolecules first of all? What is the meaning of biomolecules? What is the meaning of biomolecules? Bio means living, right? All of you know bio means living. Bio means living. And molecules means obviously all of you know the simple molecules. So biomolecules basically means living molecules. Yes, it means living molecules. And every single molecule which is present in our body, it could be in the form of carbon, in the form of hydrogen, in the form of your oxygen. All of these are biomolecules. When they combine together, they make something called as proteins, they make something called as lipids, they make something called as carbohydrate, amino acids. So all of these carbohydrates, amino acids, proteins, lipids, we will be studying in this chapter. Okay, we'll be studying in this chapter today. So students, can anyone tell me this weightage of this chapter is approximately around, let's say around usually 4 to 6 questions. 6 questions come from this chapter. 6 questions come from this chapter, and I will tell you exactly where the question will come from. So you can prepare yourself. It's very easy to understand. Right? It's very easy to understand. 6 questions from this chapter usually comes up. Now students, let's read what is given here. Right? Now students, all of you know this right here is our earth. Right? Let me tell you something. This right here is our earth, mother earth. Right? We have the earth here. Now students, on this earth there is something called as crust. Outer part is called as a crust, right? This earth has an outer layer called as a crust here. We have a crust. Earth has a crust. All of you know earth has a crust, outer crust. Now students, understand here that in this outer crust, there are certain types of elements are present. On this earth's outer crust, there are certain elements. There are certain elements such as your hydrogen, oxygen and carbon. On this earth's crust, there are certain elements such as your hydrogen, oxygen and carbon. Hydrogen, oxygen and carbon on the surface of the crust. Now students, if I tell you the same hydrogen, oxygen and carbon is also present in our body, will you believe me or not? Tell me, if I tell you the same hydrogen, same oxygen, same carbon which is found on the earth's crust is also found within us, inside our body, all of you will believe me that? Yes, sir. You'll be like, yes, sir. Obviously, whatever is present in the earth, the hydrogen, oxygen, carbon, it is also present in our body. But then what is the major difference then? What is the difference between the earth's crust components and our body's components? What is that one difference? The one difference between is nothing but the amount, the concentration. The amount of hydrogen, oxygen, carbon present in our body compared to the amount of quantity of hydrogen, oxygen, carbon present on the earth's crust. The same hydrogen, oxygen, carbon is present inside our body, but in a higher concentration. In a higher concentration. So with that being said, let's start reading what is given here. All of you. See, there is a wide diversity of living organisms in our biosphere, that is our earth, right? Now, a question that arises in our mind is, are all living organisms made up of same chemicals, that is elements and compounds? You have learnt in the chemistry how elemental analysis is performed. All of you know, right? Tell me, yes or no? Has Diksha ma'am taught you this? Has Diksha ma'am taught you this? Is it present? In chemistry, you might have studied, right? Chemical analysis, something you add on. Boom, 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 bush, you do something, right? Chemical analysis, right? You have learned in the chemistry how elemental analysis is performed. Honestly, I don't remember actually. Okay. If you perform such an analysis on plant tissue, animal tissue, or microbial paste, we obtain a list of elements like carbon, hydrogen, and oxygen, and several other and the respective content per unit mass of living tissue. Now, if we do the same analysis is performed on a piece of Earth's crust, if you take a piece of Earth's crust and you do some that magic, no? You add some chemicals, you do something. Okay. If you do the same analysis on the Earth's crust, you will obtain the same amount of same elements. See here, Earth's crust as example of non-living matter, we obtain a similar list. Now, what is the difference between the two lists? That is, they're asking you, what is the difference between living and non-living then? If living humans, the plants or the microbe also have hydrogen, carbon and oxygen, non-living things also have hydrogen, oxygen and carbon. What is the difference? I told you already. Tell me in the chat, all of you quickly. What is the difference then? What is the difference? You're giving me tacos, sir. Is that taco? Is that a taco? Okay, give me a taco, okay. Earth's crust as an example of non-living matter, we obtain a similar list. Now, what is the difference between the two lists? In absolute terms, no such difference could be made out. Absolute terms, there is no such difference. All the elements present in the sample of Earth's crust are also present in the sample of living tissue. So, same elements are present. Hydrogen is here, hydrogen is there. Carbon is here, carbon is in this. Oxygen is there, oxygen is there. Yes. However, a closer examination reveals that the relative abundance, also called as what? Also called as concentration or amount. Relative abundance of carbon and hydrogen with respect to other element is higher in any living organism than the Earth's crust. Now, if I ask you students, is there more carbon? You tell me now. Is there more carbon in this particular piece of paper, or there is more carbon in all of you? What should be the answer? Tell me. Happy Ganesh Chaturthi to you also. Tell me, students, which one has more carbon? This piece of paper has more carbon, where there is C3 cycle written here. I don't know why. Okay, it's an old sheet lying here. Okay, which has more carbon? This piece of paper or you? Tell me in the chat quickly. Obviously, you will tell, sir, living tissues has higher concentration of hydrogen, carbon and oxygen. Is the first paragraph clear to all of you? Tell me. Is there any doubt in the first paragraph? Students, let me tell you, the entire chapter is this easy. If you read a paragraph, you understand what is given, you will be able to solve any questions. Now let's go to the next paragraph. Yes, listen to Madhumati. Like all of you listen. Okay, see, look at here, students. Look at this particular diagram or chart here. If you look at the Earth's crust and human body, this side is the Earth crust, this side is the human body. If you look at this table right here, hydrogen 0.14,
0.5 in the case of human body. Carbon 0.03 in the case of human body, 18.5. Oxygen 46.6%. 65%. Do you see, students? Do you see, in the case of human body, there is high amount or relative abundance of hydrogen, carbon and oxygen. That's all. Okay, that's all. Now students, now I'll teach you something called as one experiment. Okay, now I'll teach you a simple experiment. Ready, all of you? Simple experiment number one. Okay, very simple experiment. Understand the experiment, then I'll read the lines of NCERT, then you will only tell me, sir, it is very easy to understand. Simple experiment will do, okay? Very simple experiment, okay? It's called as chemical analysis. Okay, chemical analysis. Now in this chemical analysis, I want you to give me a, let's say, should we take a rat or a plant? Tell me. Should we take a piece of a rat liver or should we take a plant? Whom should we sacrifice? You tell me whom should we sacrifice, I will tell you who to sacrifice. Tell me, whom should we sacrifice, all of you? Should we sacrifice a rat liver piece or a plant piece? Plant leaf? Tell me quickly, all of you, it's your call now. Whatever you tell, we'll take that only organism. We'll take rat, huh? We'll kill the rat. RIP rat in the chat, all of you. We'll take a rat piece and a plant piece, both, so to satisfy everyone. Okay, so we will take a liver piece of a rat. Liver. RIP rat in the chat, all of you. We can take a liver piece. All of you saying plant, huh? Oh, no, we'll take plant also. We'll take a plant piece also. We'll take a leaf also. Okay, we'll take a leaf. Also. Okay. Take a leaf or a leaf. Okay, liver. It's your choice, all of you. It's your choice. Whatever you want, you can take. Same result you'll get. Okay. Now you're supposed to take a leaf or a liver piece. This is a leaf or liver piece. Okay. Now next step is what? To make a chutney out of it. Next step is take a mortar and pestle. All of you should take a mortar, chutney making machine basically. You should take a mortar and a pestle. Okay. You're supposed to add the leaf inside. You're adding the leaf inside. Okay. You're adding the leaf in the mortar and pestle. Okay. Now, to this mortar and pestle, obviously when you make the chutney, do you add some liquid to it? Coconut chutney you're making in the house, ask your mother, she'll tell you. You're supposed to add some water into it. Now here we'll not add something like water, but we will add some solvent here. We will add some solvent here called as trichloroacetic acid. We will add what? Trichloroacetic acid. Trichloroacetic acid we'll add. We will add trichloroacetic acid. Okay, done. Trichloroacetic acid is added. Now you make a nice chutney. Nicely you grind it completely, and you will get one chutney here. Okay, and we will call this chutney as slurry. We will call this slurry. Okay. Slurry has been made. Now you take this slurry. You're supposed to now take this slurry. One second, let me divide the page. Now you're supposed to take the slurry and add it into a beaker. Okay. Okay. Now you take this slurry, and you're supposed to add it to a beaker. Let's take a beaker now. Let's take one big beaker. We have a beaker here. Okay. We have a beaker here. And on top of this beaker, I am adding a cloth. On top of this beaker, I am adding a cloth also. Here. I am adding a cloth here. I am adding a cheesecloth, also called as muslin cloth, to filter, for filtration basically. Cheesecloth or a muslin cloth. Cheesecloth or a muslin cloth. Okay. Done. Okay. Now students, you take this slurry and you pour on top of it. You're pouring it out. Okay. Done. Once you pour. Now tell me, once you pour the slurry, some amount of slurry will come down? Tell me yes or no quickly. Some amount of slurry will drip down. Some amount of slurry will be on top. When you make juice also, when you make orange juice, you're adding the juice in in a mesh, right? When you add the juice in a mesh, the liquid part will come down. The solid fibers and seeds will be present on the top. Same thing is happening. Some of the slurry will come down. Some of the slurry will be coming down. You see, some slurry is dripping down. Some slurry is dipping down. Some slurry is present on the top. Some slurry is present on the top. Okay. Some slurry is present on the top. Now, can I call this the below part? Can I call the one which is dripping down, students? One which is dripping down. Can I call this filtrate? It is filtering down. Can I call this filtrate? Filtrate. Yes. I can call this filtrate. Now, what is present on the top? Once again, let me change the color so you'll not get confusion. Let me change the color so you don't get confusion between filtrate and retentate. Can I call this filtrate? Yes, I can call this filtrate. Now, what is present on the top? The present which is present on the top here, can I call this retentate? Yes, this is called as retentate because it is retained on the top. It is retained on the top. I can call it retentate. I can call it retentate. Now, students, understand the simple concept. This particular slurry was able to drip down. Why? Because whatever is present here, can I tell it was soluble in trichloroacetic acid? Yes or no? Whichever is coming down was technically soluble in the acid. That is why it was able to come down. If it was not soluble, it would have remained on the top. So can I call the below one as acid soluble fraction? Acid soluble fraction. Yes, because the slurry which is present on the bottom is soluble in the acid. Very simple. Yes. Now, what about the present on the top? Can I call this acid insoluble? Acid insoluble fraction. Because it is not soluble in acid. Yes. Not soluble in acid. Insoluble in insoluble fraction. Okay. Okay. Acid soluble and acid insoluble. Now, students, understand. Obviously, students. Obviously, if this is able to see here, I have a muslin cloth. Imagine we have a muslin cloth here. We have a muslin cloth. In this muslin cloth, we have small grids. In this cloth, there are small grids. If there is a element or if there is a slurry which is present this tiny, can it easily pass through? Yes, it will be able to pass through easily. Now, what about we have a very large slurry? Can this very large slurry pass through the filter paper? Absolutely not. It cannot pass through because it is very large. It cannot pass through the small holes. So this pink color part will be present on the top only. So can I tell all the slurry which is present on the bottom? All the slurry which is present on the bottom, can I tell their size is very less? Can I tell, students, their size is very less? Obviously. So can I tell the size here is less than 1000 Daltons? Yes. Size is less than 1000 Daltons? Obviously. Less than 1000 Daltons. Okay. Now, what about the present on the top? Can I tell their size is more than 1000 Daltons? Size is more than 1000 Daltons. You'll be like, yes, sir, their size is more than 1000 Daltons. That's why they're present on the top. Okay, present on the top. So, can I call them, since their size is very less, can I call them micromolecules? Micromolecules. Can I call them micromolecules? Can I call them macromolecules, all of you? Macromolecules. Macromolecules. Obviously, students. So this is the simple experiment to find micro and macromolecules. And this is a simple experiment which will tell you the different types of micro and macro molecules. See this next slide. See this next slide. So what we do, students, I'll repeat again one more time. We took a RIP, this mouse, all of you. This mouse liver, we took the cut of the mouse, take the liver out. We took the liver, we took a leaf piece, we made a chutney with the trichloroacetic acid. Yes, trichloroacetic acid. No, acetic acid is CCOH, no? OH, yes. Trichloroacetic acid, yes. And then we took this slurry and we added it here. Once you added, some amount is present on the top, some is coming to the bottom. Which is present on the top is called as retentate, acid insoluble fraction, more than 1000 Daltons, macromolecules. The one which is coming down is called as filtrate, acid soluble fraction, size is less than 1000 Daltons, and they are called as micromolecules. Any doubt till here, quickly ask me in the chat, all of you. I'm looking at the chat. Your time starts now, go. Doubt part. Any doubt here? Sir, why only rat liver? Because very easy to crush now, very soft it is. So very easy to crush and lot of nutrients are there. Sony, please ask some better doubt. What is, well, I am asking if you have any doubt in the entire concept. You're asking, sir, why are you taking liver? Why are you taking only liver? Nice doubt, but yeah, nice doubt. Why only rat liver? Oh God, because it is easy to crush and easy to separate out the ingredients. See, when you're trying to crush something here, you should make sure that entire macro and micro are completely, nice chutney you're making. Okay. Oh God, crazy doubts. Now students, look at this table here. Look at this table. Why dog liver here? Dog, Papa, what did the dog do to you? Huh? Leave the dog alone. Papa, dog you want? What? Chinese you have become? What is this? Go to China. Dog you want to sacrifice. Okay. Now, molecules. What have we learned? Micromolecules is what? Size, less size, low molecular weight. Size between less than 1000 Daltons. 800 Daltons. Found in acid soluble pool. That is present on the bottom. Example is a simple sugar, students. Simple sugar. Monosaccharides, right? Amino acids and nucleotide, very small items, okay. Now, what about macromolecules? Okay, in macromolecules, large size, high molecular weight, that is size is more than 1000 Daltons, found in the acid insoluble pool, that is present on the top, also called as retentate. Example is what? Complex carbohydrates such as your polysaccharides, oligosaccharides, yes, proteins, because proteins are made up of long chain of polymers, amino acids, so, and also nucleic acid. But there is one imposter here. How many of you played that game Among Us? In the game Among Us, you always know there is something called as imposter. Imposter is always the odd one out. There is one odd one out here, students. Can anyone tell me who is the odd one out here? The one odd one out which I'm trying to bring to your attention is right here. The odd one out. The odd one out here is something called as lipid. Now, lipid is one, one exception here. Now, tell me how many of you played the game Chain? When you were in kids, obviously you still are kids, but you might have played this game, right, called chain, where you hold hands, you're by making a chain. Someone has to come and break the chain, right? So when you're making a chain, are you making a strong group together? Yes, making a strong group. Now imagine this lipid is also playing chain here. This lipid is also playing chain game. What is it doing? It is like friends. We are together, we are strong. Individual, we are very weak. So all of the lipids will come together and they'll make one entire big gang, one rowdy gang they're making. All the lipids are coming together and they're making a big gang. Now when this big gang is there, right, no one can dissolve it. So that's why, students, even though this lipids right here, the size of the lipids is less than 1000 daltons, less than 1000 daltons, okay, they will not be dissolved. Even though their size is less than 1000 daltons, lipids will be found where? Lipids will be found in the acid insoluble fraction. They'll form something called as vesicles. I'll show you the diagram later on. I'll show you the diagram also. Okay. So all the lipids will form vesicles. They'll form a big gang and they'll be like, no, I will not come down. No one can shake me. So remember students, one of the few exceptions is that lipids are found in the retentate, not on the filtrate. Even though the size is less than 1000 daltons, they'll be found in the retentate. Is that concept clear to all of you? Tell me yes or no quickly. All of you. They form something called as vesicles. Yes. Done? Done, students? Any doubt there? If there is no doubt, let's read the lines of NCERT and understand what we just learned. Okay, let's read. All of you read with me now. Everyone, don't be lazy. Today is Ganesh Chaturthi. Why are you being lazy today? See, I am coming to the office. I'm working really hard here. So I want everyone to read with me. That's how you get the actual experience. Okay, I know all of you are already diligent and you are, studying with me today also, even though it's a holiday. So read with me also. Okay, let's read. We can continue asking in the same way, what type of organic compounds are found in the living organisms? How does one go about finding the answer? How do you find an answer? What is present in living organisms? How do I know in my body I have carbon, hydrogen, oxygen? How do I know in a leaf I can find carbon, hydrogen, oxygen? How do I know? What is the answer? To get the answer, one has to perform a chemical analysis, which we just did. Okay, we can take any living tissue, a vegetable or a piece of liver, see, RIP, and grind in a trichloroacetic acid, Cl3CCOOH. Using a mortar and pestle, we obtain a thick slurry. If we were able to strain this through a cheesecloth or a cotton, here, cheesecloth or a cotton, we're doing it here, see, cotton, we would obtain two fractions. One is called as filtrate on the bottom, or more technically acid soluble pool, here called as filtrate or acid soluble pool, right, acid soluble pool, the acid soluble pool. And the second is called as the retentate or the acid insoluble fraction. Scientists have found thousands of organic compounds in the acid soluble pool. Many can be found. Okay. Now, in higher classes, you will learn how to analyze living tissue sample and identify a particular organic compound. It will suffice to say that here one extract of the compounds, then subject the extract to various separation techniques till one has to separate a compound from all the other compound. Basically, higher filtration methods. Okay. In other words, one isolates and purifies a compound. Analytical techniques, when applied to compounds, give us an idea about molecular formula and probable structure of compounds. So they are trying to tell you is what you do here, right? Once you make the separation here, right, you can take this slurry, add it to machines, and the machine will tell you which particular organic compound is present there. And that's how you will know carbon is present, oxygen is present, and hydrogen is present. So this is just the first step. The next step is going to be the other advanced analytical techniques. Sir, what is the function of trichloroacetic acid? Trichloroacetic acid helps in the chutney making process. The main function there is to open the cell wall and break the plasma membrane. See, unless you break the cell wall and plasma membrane, do you think the cell contents will come out? No. The cell contents will remain inside only. So if you want to break the cell level, you want to break the plasma membrane and the cell wall in the case of plant. So trichloroacetic acid does that function. It does that function. Okay? Very simple. Now, living organisms also have also got other inorganic elements, students, inorganic elements. So the first experiment gave us some idea about the organic molecules, right? Organic molecules. Now can we do one separate experiment to find if plants have inorganic molecules also? Let's do a simple experiment now. Yes. Let's do a simple experiment to find the inorganic molecule. This is your experimental number 2. Experiment number 2. Okay, let's start. Second experiment. Okay, second experiment right here. Now, second experiment is even more simple. It is called as Ash test. It is called as Ash test. Not your Pokemon Ash. Ash is the powder. Okay, Ash test. Very simple again. Again, you can take let's take a leaf only this time. Like a leaf. We have a leaf here. This is a leaf. Obviously, we have a leaf here. Now students, will this leaf has water inside? You'll be like, yes sir, leaf has water. You can tell me this is a fresh leaf. It has water inside. So can I call this fresh leaf? This is your fresh leaf. Fresh leaf has something called as wet weight. Wet weight. Obviously it will have wet weight. Obviously it will have something called as wet weight. Why? Because water is present. Because water is present here. Water is present here. Right. Now you take this leaf and you evaporate it. You do evaporation. You do evaporation. Once you do evaporation, students, once you do evaporation, can I tell? Water is being lost. Water is being lost. Yes. Now we have something called, we have one very sad looking leaf now. Without all the water, we have one sad looking leaf. Very sad it is. This sad looking leaf is called as what? It's called as a dry leaf. It's called as dry leaf. Can I tell in this dry leaf, right? This dry leaf will have something called as dry weight now. Dry weight. Dry weight. Yes, it has something called as dry weight. It has the dry weight. Now you take this dry weight leaf and you completely grind it. You can take this thing only. What is that? Mixie. Take a mixie, nicely grind it. All of you have mixie in the house now. Imagine you have a mixie in front of you. In that mixie, you are adding all the leaf and you're nicely grinding it. Nicely grinding it. Now, once you make the when you make the grinding, will you get powder? Yes, we'll get the powder. Now you completely burn the powder now. Also called as incineration. Also called as what? Incineration. Completely burn the powder. Now students, when you burn the powder, what happens? When you burn the powder, we get the remaining is called as the ash. Remaining is called as the ash. So when you burn, students, when you completely burn it, remaining carbon is being lost. Remaining water, also if some amount of water was there, everything is being lost. Can I call this oxidation? Oxidation is happening. Oxidation is happening. Now you can take this ash and you can subject it to certain chemical analysis. Chemical analysis, and you take this ash and you do chemical analysis, you will obtain the information regarding the inorganic compounds. You will get information regarding the inorganic compounds. Inorganic compounds present. Compounds present. This is called as the ash test. This is called as the ash test. That's all. That's all. Okay. Let's see what is given in NCERT now. Let's see what is given in NCERT. A slightly different but destructive. Why destructive? Chick chick boom. Fire you're adding, right? Fire you're adding. That's why destructive method. Okay. Destructive experiment has to be done. One weighs a small amount of leaf tissue, say a leaf or a liver called as wet weight, and dry it completely. All the water evaporates. The remaining material gives dry weight. Now if the tissue is fully burnt, burn all the carbon compounds, oxidation. Carbon compounds are oxidized to gaseous forms, that is CO2 and water vapor, and are removed. What is remaining is called as the ash. What is remaining is called as the ash here, ash. Remaining is the ash. This ash contains inorganic elements like calcium, manganese, etc. Inorganic compounds like sulfate and phosphate are also seen in the acid soluble fraction. So if I ask you a question, students, if I ask you a question, name the inorganic elements. Name the inorganic elements found in the acid soluble fraction. What will be your answer? Tell me. So when will you do class 12 chapters? Whenever we want. I have done few chapters. Class 12th, principles of inheritance is done. Right? We have finished all that chapters. Human reproduction is done. Sexual reproduction of flowering plants is done. Right? Almost all class 12th is done. Don't worry. This is just the NCERT revision now. Now students, tell me. If I ask you a question, which elements are found in the inorganic elements? Inorganic elements are found in acid soluble fraction. Acid soluble fraction, what will be your answer? All of you in the chat. Answer should be what? Sulfate and phosphate. Not manganese and calcium. Sulfate and phosphate mainly. Okay. See, this is calcium and magnesium is found in your ash test. But the one more line is your important line of NCERT which everyone ignores. Inorganic compounds like sulfate and phosphate are also seen in the acid soluble fraction. So remember students, sulfate and phosphate are also found in acid soluble and which are inorganic elements. Inorganic elements. Okay. Therefore, elemental analysis gives the elemental composition of living tissues in the form of hydrogen, oxygen, chlorine and carbon. That's it. While analysis of compound gives an idea of the kind of organic, okay. From chemistry point of view, this should ask Diksha ma'am, okay. Chemistry point of view, one can identify functional groups like aldehyde, ketone. Students, what is the functional, how do you write an aldehyde? I don't remember, tell me. How do you write an aldehyde? How do you write an ketone? Tell me in the chat, all of you. How do you write an aldehyde and how do you write an ketone? In the chat, all of you. If you don't write, I'm complaining to Disha ma'am. Now I'm telling you, I'll complain. Okay. Aldehyde, ketone and aromatic compounds, etc. But from a biological point of view, we shall classify them into amino acids, nucleotide bases and fatty acids. That is what we'll study in today's class. We will study something called as nucleotides. We will study something called as fatty acids. We will study something called as amino acids. Okay. This is the elemental composition of inorganic compounds found in the ash test. Okay. List of representative inorganic constituents of living tissue. Okay, living tissue has all of this. COO is an aldehyde. Okay. What about ketone? What about ketone? Tell me. Ketone is CHO is aldehyde. Well, very easy. If you know the structure of glucose, you can tell. What is ketone then? How do you write ketone? Ketone. CO. Okay. Now students, ready for amino acids now to learn amino acids? Tell me students, are you ready to learn amino acids now? First part of the chapter is over actually. First part of the chapter is actually over now. Tell me if you're ready to learn amino acids now. Amino acids. C double bond O. Exactly. C double bond O is your ketone. Yes. Tell me, till now is it easy class or not? Tell me, till now whatever you learned, the two experiments, super easy, right? Students, let me tell you one more time. Let me tell you one more time today. I'll keep reminding you this. This NCERT line by line, if you notice on our channel right now, every single session is almost 3 hours. Right? Now, for example, if you take photosynthesis in higher plants, which you just finished yesterday, I have done photosynthesis in higher plants for 6 hours. 6 hours I have done the class. I can finish the entire chapter if I teach in the RNA series. I have finished the entire chapter in just 2 hours, 40 minutes in RNA and 6 hours in NCERT. 6 hours in NCERT. Why do you think so? What is the major difference between 2 hours 40 minutes in RNA and 6 hours in NCERT? The major difference is that, students, when you read from NCERT line by line, yes, it is going to be a longer session. But when you do from NCERT, there is no way that you will miss out a single line of NCERT. And you know, there can be a question from any part of the chapter. It could be a diagram, a legend, or any small hidden line. For example, we just now learned, see, usually this line here, which everyone ignores this line here. See here, everyone ignores this line. Inorganic components like sulfate and phosphate are found in both acid soluble fraction and also they're inorganic in nature. So this line can be easily missed out sometimes. But when you do NCERT line by line, right, line by line if you do, trust me students, you will have the ultimate confidence. And in NCERT line by line, you might have noticed, after every class, when you're finishing a class, do we not do a lot of questions? After every class, I make sure my students solve enough of PYQs to get the confidence back, right? So that is the meaning of the detailed lectures. And honestly, I'll tell you, we cannot do this when the exam is coming closer. You absolutely cannot do this. So this is my advice to all of you. Now is the time. Take the advantage of the time right now given to you. Don't waste your time on unnecessary things thinking that during the last phase of my examination, I will study everything and I will become topper that time. No. Rome was not built in a day. A topper cannot be made in a day. A dropper can be made in a day. So take your time right now. You have the time and luxury right now. Use it to do a long sessions with us. Enjoy every line of NCERT. Understand. Question us. Tell me, students, have I not ever answered any doubt in the class? Obviously. Every single time you ask a doubt, I always reply. To make sure whatever you learn in the class is already learned to you. You do not have to revise it again later on 10 to 15 times. Yes, you need to revise. But once it is done in the class, just revise the same day and you're sorted. Revisit the chapter after one or two weeks. Again you revise. Take advantage. Take advantage of what is given to you. Don't take it lightly. Don't take it lightly right now because you have the luxury of time right now. Don't later on cry telling that, sir, I'm not able to, haven't read the NCERT 2 times, 3 times. And I've told you before, read the NCERT at least 5 times before your examination. 5 times minimum. Minimum to minimum 5 times. With me you're studying at least 1 time. On your own you're supposed to study at least 4 more times. That is the resolve which you need to have. Okay. So understand what I'm saying. Now we will do amino acids. Amino acids. The reason we are going so detailed for you to understand is because you don't have to waste your time later on. We do not want you to even think anything else. No distraction at this point. Okay. Now tell me, amino acids. Amino acids. Do you find amino acids in the acid soluble or acid insoluble? Tell me the check quickly. Tell me, where do you find amino acids? In retentate or filtrate? Tell me. If you go back here, this diagram which I showed you, I told you here, amino acids are size very less. Amino acids are present as a biomicromolecules. Micromolecules. Micromolecules are present in the filtrate in the acid soluble pool. Right? Acid soluble pool. So can I write for amino acids? Amino acids are micromolecules. 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 Their size is less than 1000 daltons. 1000 daltons. Yes. They are found in acid soluble pool. They are found in acid soluble pool. Yes. Acid soluble pool. They are a part of filtrate. Part of filtrate. Now students, tell me, when many amino acids join together, when many monomers come together, what do we get? When many amino acids come together, when all the monomers join the hands, we end up getting the proteins. We end up getting the proteins. Clear? All of you clear with this? We end up getting the proteins. There are 2 types of amino acids, students. There are 2 types of amino acids. All of you understand. There are 2 types of amino acids. There are 2 types of amino acids. Understand? 2 types. One is called as essential, other one is called as non-essential. One is called as essential, the other one is called as non-essential. Essential and non-essential, what is the difference? Tell me. There is something called as essential. Essential and non-essential. Non-essential amino acids. Tell me, what is the English meaning of essential, students? What is the meaning of essential? Essential means important. Essential means important for our body. And since it is very important to our body, even though it is very important, our body cannot make it. It is not produced by the body. Not produced by the body. If it is not produced by the body, how do we get it? Obtained by food. So every single time you eat food, you take up the essential amino acids. Essential amino acids. And all of you who go to gym, you have seen them, people who go to gym drinking protein powder, right? They take this whey protein and they drink whey protein powders. The whey protein basically has the essential amino acids. Essential amino acids. Now what is non-essential? Produced by the body. It is produced by the body. It is produced by the body. Yes, it is produced by the body. That is basically non-essential. Non-essential. Now students, tell me what is a methane? Can anyone tell me what is a methane? Structure of methane in the Go chat. I want everyone to be active. Do not be lazing around right now. Tell me what is the structure of methane, all of you. Methane structure. Methane. Structure of methane. Structure of methane, all of you in the chat. Is it CH2, CH3, CH4? Methane is CH4, right? So, can I tell this right here is a structure of a methane? Yes. This right here is a structure of methane. Yes. Structure of methane. Okay, structure of a methane. Now, amino acid, listen to my words. Amino acid is nothing but a substituted methane. You take a methane like this. You take a methane and you substitute its part. You substitute its part. For example, this particular hydrogen is replaced by COOH, carboxylic acid group. Carboxylic acid group. This H right here is replaced by a amino group. Amino group. Amino group. Amino group. This hydrogen right here is replaced by a functional group. It is replaced by a functional group, and that is how we get an amino acid. We get an amino acid. This right here is a substituted methane, and this right here is an amino acid. Amino acid. For example, now this R, students, this R can vary. This R can vary now. Understand? Understand. Let's draw it. Let's draw it one more time. So we have carbon in the center, COH, carboxylic acid this side, amino group this side, amino group. Here we have hydrogen. Now here we have R. Now students, this R can be anything. This functional group can vary. This functional group can vary. Right? This functional group can vary. Right? Functional group can vary accordingly. Okay. Functional group can vary accordingly. For example, this R can be present as hydrogen. This R can be present as hydrogen. Okay. Now, what about if we make the R as a methyl group? It can be present as methyl group. Okay. Or it can be present in the form of hydroxymethyl, that is CH2OH. CH2OH. Yes. CH2OH. This R. When the R group, the functional group, students, when the R group or the functional group is present in the form of hydrogen, we call this glycine. We call this glycine. We call this glycine. Glycine. All of you write down in your book right now. Write down. When only hydrogen is present, it's called as glycine. When it is present, when we have a methyl group, when we have a methyl group such as CH3, it is called as alanine. Alanine. Now, last one, friends, last one. When the functional group, the R group is present as a hydroxyl methyl, it is called as serine. It is called as serine. It's called as a serine. Okay, it's called as a serine. This is the 3 different amino acids given in your NCERT. Yes, they can ask you question on this. Yes, they can ask you question on this. Let's go here and see that. It's not given here. Where is it? Where is it? Where is the diagram? I've added the diagram somewhere. I've added the diagram. Can you see it, students? Can you see it's given in NCERT? We have the carboxylic part, carboxylic group, amino group, right? NH2 group, and we have the R group. When the R group has hydrogen, it's called as glycine, methyl alanine, and hydroxymethyl, it's called as serine. These 3 you need to know. These 3 you need to know. Everyone in the chat, please write down your book right now and mark all of this, so because this will come in handy later on. Glycine, alanine and serine. 3 of them done and dusted. This is your homework for today. Once you finish your class with me, open your book. Yes, open your book. Write these 3 different structures at least 3 times and send it to me in Telegram channel. And all of you know the Telegram channel, right? This is your homework. I will check your homework. You can send the Telegram channel. Small homework for all of you. Write it 3 times and show it to me. Okay. Otherwise, punishment is there. Okay. Now done. That was basically the amino acid. Now, apart from here extra points I will add for glycine. Here glycine is basically non-chiral in nature. Now, what is the meaning of chiral carbon, students? What is the meaning of chiral carbon? Chiral carbon is a carbon which has 4 different groups. For example, here there is a carboxyl, amino, hydrogen and methyl group. 4 different groups, it is a chiral carbon. Alpha carbon this is alpha carbon, it's a chiral carbon. But in the case of glycine, students, in the case of glycine, don't we have H here and H here? Yes, sir. So can I call glycine as non-chiral? Can I call this as non-chiral in nature? Non-chiral in nature? Yes. Because it is non-chiral and it has a simple H here, simple hydrogen, can I call this simplest amino acid? Simplest amino acid. It is the simplest amino acid, non-chiral, and it is optically inactive. Optically inactive. Optically inactive in nature. Yes. Non-chiral in nature. Because they have hydrogen on top, hydrogen bottom. A chiral carbon will have 4 different functional groups. But in the case of glycine, hydrogen on top, hydrogen on bottom, and that is why it is non-chiral in nature. Done. Understand that small part. Now, students, there is something called as aromatic amino acid. All of you know basic chemistry, right? Aromatic means the benzene ring is present. In the case of phenylalanine, tryptophan and tyrosine. Phenylalanine, tryptophan and tyrosine, there is an aromatic benzene ring attached, and they are called as aromatic amino acids. Diagram is not important, names are important. Remember the names. So write also this in your book and show it to me. 3 different types of amino acids are aromatic in nature. Now, apart from that, students, you need to understand amino acids can be 3 types again. Amino acids can be 3 types. For example, an amino acid can be acidic in nature, it can be basic in nature, or it can be neutral. Doesn't need any problem in life. Acidic, basic and neutral amino acid. For example of acidic, example is very important. In examination, they will ask you directly example, they will not ask you the structure. This structure they will not ask you. They will only and only ask you the example of acidic amino acid, example of basic amino acid, and example of neutral amino acid. Can anyone tell me? Students, acidic amino acid, remember, there is aspartic acid and glutamic acid. Aspartic acid and glutamic acid, it name only has acid, obviously it is acid then. If the name only has acid, it is an acid. So remember, aspartic acid and glutamic acid are acidic amino acids. How do I know, sir? Look at the structure of it. Structure not important. Only to learn it is important, but examination they will never ask you this. Okay. Look at aspartic acid. It has the carboxylic group here and it has an extra carboxylic group here. So if there is an extra carboxylic acid group, can I call this acidic in nature? Yes, it is acidic in nature then. Acidic in nature. If you look at the glutamic acid, it has a carboxylic acid group here. Here also it has a carboxylic acid group. So can I tell glutamic acid and aspartic acid are acidic in nature? I want every single student to type in the chat the two examples of acidic amino acids. Now students, basic amino acid. In the chapter molecular basis of inheritance, you might have read this actually. The basic amino acids are three of them. Lysine, arginine and histidine. Lysine, arginine and histidine are the three basic amino acids. I want everyone to tell in the chat. Everyone to type this in the chat, and if you're watching it recorded, I want you to repeat after me. The three basic amino acids are lysine, arginine and histidine. Three basic amino acids. Why, sir? Why do you call them basic amino acid? Because they have an additional amino group. Apart from the main amino group, they have an additional amino group. Here also, can you see? Additional amino group. Additional amino group. NH2 group is extra. That's why they're basic in nature. That's why they're basic amino acid. Basic amino acid. Okay. Basic. So remember students, basic, acidic, aromatic, as well as these three different examples, and you are good to go. No one can take any marks from you. That's all. Now, neutral amino acids. In your NCERT, there might have mentioned other name, but I have written glycine here because the diagram is very easy to understand. But understand there are 15 other neutral amino acids. Now, students, understand this simple thing. There are total, so tell me in the chat how many total amino acids are there? Can anyone tell me what is the total count of amino acid? The total count of amino acids are 20. Right? Total amino acids are 20. Okay. Amino acids are 20. Out of these 20 amino acids, two are acidic. Aspartic acid, glutamic acid. Two are acidic in nature. Acidic in nature. We just now learned 3 are basic in nature. Basic in nature. Remaining 15 are neutral. 15 are neutral in nature. Neutral. So that is how much? 20 amino acids. 20 amino acids. 2 acidic, 3 basic and 15 neutral amino acids. Easy. Will you remember this now, students? Yeah. Amazing, Gopika ma'am. AG, aspartic acid, glutamic acid. Right? Nice. 15 neutral, all of you. Now, let's read what is given you in NCERT. Before we go, do you want to learn what is deuterion? Students, before we read NCERT, should I explain what is deuterion now only? Do you want to learn? What is deuterion now only you want to learn? Or do you want to learn after reading NCERT? It is very easy to understand. Deuterion is very, very easy to understand. There is nothing complicated about it. Nothing complicated about it. I'll teach you deuterion, you will learn it very easily. Want to learn now only? Okay, beautiful. So let's understand what is deuterion. Now, if you want to understand what is the meaning of deuterion, you need to understand that as the amino acid is like all of you. The amino acid is like all of you. You tell me. If you're sitting in the class, and you get a news that the maths teacher is absent today or the physics teacher is absent today, you're suddenly happy. No physics class today. And then you're thinking that the substitute teacher is games period. Now tell me, if you get games period when the physics sir is absent, will you be happy? Or you will be like, oh, full happiness, you'll start jumping around and run to the ground. But imagine the physics sir is absent, but the class is taken by chemistry ma'am, a chemistry sir. You will again be sad, ayo, physics sir is absent, chemistry ma'am came off. Now tell me, did you react differently? Did you react differently to getting a games period and getting a extra chemistry period? Was there a change in difference? Yes. Right, there is change in difference. So how you react to the surroundings? How did you react to the surroundings? Similarly, even amino acid will react differently to the surroundings. Amino acid will react differently to its surroundings. Every single time amino acid is in acidic medium, it is acting very weird again. Every single time an amino acid is in a basic medium, it is again acting very weird again. So let's understand, how does an amino acid react with acid? How does an amino acid react with a base? Only when you understand that, you can master this vitrion very easily. Ready? Listen with me, all of you. What exactly happens here? What exactly happens? Now let's draw a simple amino acid. All of you draw the simple amino acid. What do we have, students, in amino acid? In amino acid, we have the carbon, carboxylic group, a amino group, a hydrogen, and a functional group. You take this amino acid and you keep it in acidic medium, full burning acid in acidic medium. Once you keep in acidic medium, what happens? Friends, remember, in the acidic medium there is H plus is present. H plus is present throughout. Yes, H plus is present. This H plus will be taken up by this NH2 right here. This NH2 right here, and this in the acidic medium. In the acidic medium, this particular amino acid will turn into basic. NH3 will become, and H group. It will become basic in nature in acidic medium. Okay. Now what about an amino acid? What about an amino acid which is in basic medium, a strong base? If you keep it in basic medium, what will happen? If you keep it in basic medium, what will happen? What will happen if you keep it in a basic medium? When you keep it in basic medium, students, when you keep this in basic medium, this H is lost to the surroundings. So we end up getting what? NH2, carbon, COO minus, hydrogen, and R. So can I tell this has become acidic in nature now? Acidic in nature. Yes, it has become acidic in nature. Now this shows that a particular amino acid reacts very differently to acid or to a base. But sometimes what happens, students? Maths teacher never absent. Maths teacher, maths class, I'm not going to comment anything on that. Very critical to comment on maths class. What if? Now students, sometimes what happens is, we have a particular amino acid here. We have an amino acid here. Sometimes what happens is, this particular proton, instead of going to the surroundings, is taken up by NH2. It is taken up by NH2. Okay. Now this is called as intra-proton transfer. This is called as intramolecular. This is called as what? This is called as intramolecular proton transfer. Intramolecular proton transfer. proton transfer. So when this happens, you will end up getting something like this. This will become H3 plus and NCOO minus and hydrogen and R. Students, this particular structure right here, as a result of intramolecular proton transfer, it's called as amphoteric ion. It is called as amphoteric ion. Amphoteric ion. This amphoteric ion is also called as what? It's also called as your Zwitter ion. It's also called as Zwitter ion. It's also called as Zwitter ion. It's also called as Zwitter ion. Now, this Zwitter ion, students, this proton transfer, will it happen in any pH? Tell me, this will happen in any pH? No. It will never happen in any pH. This intramolecular proton transfer will happen in a only particular pH. In a very specific pH. Very specific pH. That pH is called as what? The pH is called as isoelectric pH. Isoelectric pH. Isoelectric pH. Isoelectric pH. Isoelectric pH. Now if you can see here, students, the diagram given here. A, first you see the A. What is happening in A? This is becoming NH3, this is COOH, right? NH3COH, what is that? This NH3COH is right here, right? That is, it is becoming basic in nature. Why? Because it is kept in acid media. This is kept in, if you keep it in acidic media, you'll get this. If you keep it in basic media, you'll get this, CO minus. You'll get this in basic media. But if you keep it in a isoelectric pH, you will get a Zwitter ion. That is, B is called as the Zwitter ion. Zwitter ion is a temporary state. It's a temporary state in which net charge is 0, students. Here, understand, a molecule with one functional group having positive charge. One side there was a positive charge, other side there was a negative charge. Positive charge and negative charge both are present together. Here. Can you see? And other functional group is a negative charge. So net charge is always, that is, positive charge is equal to negative charge. Net charge is 0 here. So in a Zwitter ion where positive charge and negative charge are present together, here the net charge is 0. Such particular ion is called as Zwitter ion. And this Zwitter ion is formed at a particular pH. The pH is called as isoelectric pH. Tell me, all of you understood? Did all of you understand? Sangeeta, Savita, Soni, Divya. This is called as isoelectric pH or something called as Zwitter ion. Done? Yes. This is called as Zwitter ion formation. Very easy to understand, students. Very easy to understand Zwitter ion. Now, let's read the lines of NCERT and let's see if we covered every single point or not about amino acids. Let's go to the top. And start studying NCERT lines. See here, what is given in NCERT? Amino acids are organic compounds, right? Organic compounds. We got to know that it is an organic compound with the help of your the first experiment, right? Containing an amino group and an acidic group are substituted on the same carbon. That is the alpha carbon. That is, this is the one carboxylic group and one amino group, hydrogen and R. This is the alpha carbon. Okay. They are substituted methane. See, written right here in your NCERT. Substituted methane. Okay. There are 4 substituted groups occupying the 4 valence positions. So, students, what is this valency? Why there is 4 valency? Can anyone tell me? Chemistry in the chat, tell me. I will show you to Diksha ma'am later on. Okay, 4 valence position. Now these are hydrogen, carboxyl group, amino group and a variable group designed as R. Based on the nature of R group, there are many amino acids. However, those which occur in proteins are only 20. There are only 20 amino acids. There is only 20 amino acids. Okay, 20 amino acids. The R group in this proteinaceous amino acid could be an hydrogen. When it is hydrogen, it is called as glycine. A methyl group, it is called as alanine. And hydroxymethyl group, it is called as a serine. Called as serine. So all of them is given one by one. Is given by one. Yes, obviously blackmailing. Obviously, what is life without blackmailing? It's called as emotional blackmail. It is not normal blackmail. It is the emotional blackmail which your mother also does sometimes when the work needs to be done. When the work needs to be done. This is white mail. Okay. There are 3 out of the 20 amino acids shown in figure 9.1, which I showed you later. So remember alanine, serine and glycine. The chemical and physical properties of amino acids are essentially of amino acids, carboxyl and R functional groups. Based on the number of amino and carboxyl group, there can be acidic. Friends, remember I told you, in acidic amino acid, there is more carboxyl group. In basic amino acid, there is more methyl group. Simple, right? There is more amino group. There is more amino group. Right? Now, acidic amino acid is glutamic acid. The base, that is lysine, is the basic amino acid along with histidine and arginine. And neutral, they are given you valine here. There are 15 other. Amino acids. Similarly, there are aromatic amino acids, tyrosine, tryptophan and phenylalanine. A particular property of amino acid, like how all of you are happy in games period, sad in chemistry period, okay, or physics period. A particular property of amino acid is ionizable nature. That is, it can take up a charge and positive or negative charge, right? Ionizable nature of NH2 and CO groups. Hence, in a solution of different pH, so in acidic pH or basic pH, the structure of amino acid changes. With time amino acid is changing. With the time, you should also, all of it should change now. Become more serious. Exam is coming near. Do you know how many days are left? Students, do you know how many days are left right now in examination? I think approximately you have around 230 days or something. 30 point some days. Right? So be serious right now. Right? Changes with solution. So that is the end of amino acids now. That is the end of amino acids. Any doubt in amino acids, students, tell me. Maths and biology, yeah. Maths is 239 days. See, 239 days you have. So change the time now. Time to change in life. Become like amino acid. You always should become a zwitterion now. Okay. So with that, amino acids is over. Students, in today's class, we will cover lipids. Okay. After lipids, I will teach something called as secondary metabolites. And I'll also teach you something called as macromolecules. One small page is there. That's it for today's class. That's it for today's class. You can go celebrate a festival, do whatever you want. Run around in the house or do whatever you want. Okay. Ravindra, any serious angry face. I don't know why the angry emoji always makes me laugh. I don't know why. Okay. 239 days to go. So students, all of you should be aware, right? All of you should be aware. Do you have a proper timetable? Let me ask all of you how you're doing first of all. Okay. All of you have a proper timetable with you, right? Yes? Tell me whether you're finishing your syllabus, all of you in the chat. All of you recording also, you can tell me in the comment section. By which month you are planning to finish your entire syllabus? Don't tell anything after January. Because your entire syllabus, at least once or twice or thrice, depending upon you, should be over maximum to maximum by December. By December. Okay. 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 By December, your entire syllabus should be over. Whatever you say, whatever you do, how much you are crying you do, festival you do, anything is fine. But by December, at maximum to maximum, your syllabus should be over. After that, none of you should be like, sir, I have not done my syllabus. Because students, in the month of January, February, March, especially January and February, you will see a big change in our channel. In the month of January and February, you will see a major change in the channel, and you will be part of it. All of you will be part of it. You should be ready by then. All of you should be ready by then, because in January and February, the amount of classes which we will take for you, you will be tired. Because that's a very serious time, right? It's do or die that time, do or die time, right? In the month of January, February, the amount of revision we will make you do, the amount of question practice will make you do. We will make sure you solve so many questions, you will be super tired. You're like, no sir, I cannot do more. Until you say that from your own mouth, we'll keep on solving you questions. When the month of March, April, dedicated test series, or any doubt you can always contact us. And by then you will be super ready with your preparation. So light that fire inside you and be ready for what is going to come to you. Because right now, this is just the phase when you do the preparation. Detailed study you're doing. Detailed. See, every single chapter you're going line by line and you're preparing. But as soon as December ends, we'll pick up the pace here in the channel. We will do short videos. We will cover the entire chapter in 1 hour, 2 hours. I will cover this entire chapter in what, like a maximum in 2 hours, I'll finish the entire chapter only. That time you should not cry, sir, I'm not able to follow, I'm not able to complete my syllabus. Okay? So this is the time when you ask the doubts, when you clear your detailed structures once, detailed study. So be serious. Why am I telling you all this is because I feel some of you are become reluctant. I saw it in yesterday's class, right? Photosynthesis and higher plants is such an important chapter, students. And I saw many students did not attend that class, many students did not appreciate how much effort is put into in the video. Because I could not see any comments at all. So that made me realize, yes, I have to give you a warning, all of you. A nice warning should come to all of you. Right? Right? A warning should come to all of you. This is the warning, wake up call for all of you. Okay. Now students, let's do lipids. Let's do lipids, all of you. Lipids. Let's do lipids now. Tell me students, lipids, all of you know, lipids are basically organic compounds. They are organic compounds. Yes, organic compounds. Yes, they are organic compounds. Yes, organic compounds. Now tell me students, what is their molecular weight? Tell me, is their molecular weight more than 1000 daltons or less than 1000 daltons? All of you tell me in the chat. Everyone. The molecular weight of lipids, can I tell it is less than 1000 daltons? Less than 1000 daltons. No always chilling. There is no point in chilling right now. Okay, only seriousness. Less than 1000 daltons. Now tell me, will you find them in acid soluble or insoluble then? Even though the size of lipids is less than 1000 daltons, you will find them in acid. You will find them in acid insoluble fraction. Insoluble fraction. That is on the top. Acid insoluble fraction. But one important point to be noted here is that unlike your monomers, when they join together, unlike monomers, they don't find polymers. They are not polymeric in nature. They are not forming polymers actually. They're not forming like polymers, right? They're not forming polymers. They are actually making a vesicle. I'll show you a diagram. You'll understand. They're forming a vesicle. Okay. Now in your NCERT, entire lipids is mainly divided into two categories. That is simple lipids. Your simple lipids can be classified into something called as fatty acids. Fatty acids and glycerol. And glycerol. And glycerol. Now what is fatty acid, what is glycerol? I'll tell you in the next few minutes. Understand. Students, this is what I was telling you. Your entire lipids will form a chain. You see, this is called as a vesicle. You might have studied this in lipid bilayer. Lipid bilayer in the plasma membrane structure. Right? This is vesicle. Since they are forming vesicle like this, they cannot pass through the muscling cloth. They cannot pass through the muscling cloth. Okay. Now students, let's try the difference between the two types. Let's try the difference between what is a fatty acid and what is a glycerol. What is a fatty acid and what is a glycerol? All of you in the chat. Glycerol and fatty acid. First we will do fatty acid. First we will do fatty acid now. Fatty acid, okay. We'll write fatty acid now. Fatty acids. Fatty acids. Now what is a fatty acid, students? Fatty acid is nothing but it is a group of carboxylic acid in a functional group. It is nothing but a carboxylic acid. It is a carboxylic acid plus the R group. Plus the R group. That's all is a fatty acid. For example, we have COH, carboxylic acid part, and we have a functional group. This right here is the carboxylic acid part. Here is the functional group. Here is the functional group. This R, students, remember, this R right here, can be a methyl. Yes, this R right here can be methyl. It can be a ethyl group, or it can be any carbon between 1 to 19 number. Any carbon number between 1 to 19. That is CH2 attachment. CH2 attachment can it be? Okay, that is basically a fatty acid. Fatty acid. Okay, and this fatty acid can be actually of two types. This fatty acids can be of two types actually. Fatty acids can be of two types. One is called as saturated, other one is called as unsaturated. Saturated and unsaturated. What is the difference? Understand, your saturated will be made up of only a single bond. It will be made up of a single bond. While your unsaturated fatty acids are made up of double and triple bond. Double and triple bond. Double and triple bond will be here. Double and triple bond. Your saturated fatty acids will have the melting point will be very high and they are solid at room temperature. They are solid room temperature. Now what about unsaturated fatty acids? Their melting point is very low and they are liquid at room temperature. Now how do you remember this? Very difficult to remember, simple. Every single time, students, tell me, which type of dosa do you like? Which type of dosa do you like? Do you like masala dosa? Do you like onion dosa? Do you like butter masala dosa? Which one do you like? Tell me. Which is your favorite? My favorite is always benne masala dosa. My favorite is benne masala dosa. That is basically butter dosa. And I always remember butter dosa is always makes me satisfied and is saturated. It makes me saturation. It gives me saturation always. It gives me saturation. Butter masala dosa. Okay, gives me saturation inside me. I'm like, I'm full. Butter masala dosa. So butter, it can be made into ghee. Yeah, it can be ghee. And ghee, have you noticed? In a room temperature ghee, unless you heat it up, tell me, is ghee solid or is it liquid at room temperature? Tell me. Tell me. Any dosa is super. Onion dosa. Okay. Tell me, is ghee solid or liquid at room temperature? You might have noticed if you open a ghee box, it is very solid. Once you heat it, it will become liquid. Yes. So example here is butter or ghee. Butter or ghee. Remember students, when you try to fry something, if you add butter, it takes more time to become hot. But if you use some oil, sunflower oil, cooking oil, will it become hot very fast? Yes. Hot becomes very fast. And you might have noticed the sunflower oil, right? The sunflower oil or the cooking oil basically. The cooking oil is always liquid at room temperature. Yes. This cooking oil is always liquid at room temperature. Cooking liquid at room temperature. when you add the oil in the frying pan, liquid room temperature. So that is the difference. That is the difference between saturated and unsaturated. Here one example will come called as gingerly oil, which I will show you later in the NCERT line. Gingerly oil. Example. Gingerly. Sounds very stupid. I don't know who keeps the name as gingerly oil. Gingerly. Sounds only very funny. Gingerly oil. Okay, gingerly oil will come in unsaturated fatty acids, we'll tell you later. Gingerly oil. Okay, so that will come in the unsaturated part here. Okay, it will come, eventually it will come. Okay, now students, now we will learn something called as glycerol. About glycerol. Glycerol. Okay, glycerol. Okay, glycerol. Now what is glycerol? Glycerol is nothing but an alcohol, students. Glycerol is nothing but an alcohol. Glycerol is nothing but an alcohol which is made up of 3 carbon, 5 hydrogen. It is made up of 3 carbon, 5 hydrogen. 5 hydrogen plus 3 hydrogen. So once again, structure just 3 hydrogen and 5 OH plus 5 OH. And understand the diagram. 3 carbon, 1, 2, 3. How many OH? How many OH, students? 3 OH. 3 OH. What I wrote? Diagram. I'll write the diagram first. Wait. Then it'll be easy to write. We have OH here. We have OH here. And we have OH here. So 3 carbon, 3 OH, and 1, 2, 3, 4, 5. 5 hydrogen. And 5 hydrogen. Okay. Okay. This is the structure of your glycerol. Tell me students. 3 carbon, 1, 2, 3. In the terms of chemistry, what do you call 3? Tri. Can I call it tri? Tri. What do you call OH as alcohol? Can I call this hydroxyl? Hydroxyl. It is an alcohol with 3 carbon. 3 carbon. Trihydroxyl because 3 different OH. 3 carbon. Can I call it propane? Can I call it propane? Trihydroxy propane. Yes, trihydroxyl propane. Can I call this trihydroxyl propane? Trihydroxypropane. 3 hydroxyl groups, the OH, hydroxyl propane. Why 3 carbons? Yes, trihydroxyl propane. Trihydroxyl propane. Students, how many of you have gotten any time cut or ulcer in your mouth? How many of you gotten cut or ulcer in your mouth? If you get ulcer in your mouth, they ask you to add glycerine, right? Glycerine they ask you to add glycerine. Have you wondered? Right? Glycerine. Tell me students, that glycerine, is it colorless? It's kind of sweet little. And it is very viscous. It's very viscous. That is nothing but is here. Glycerol is nothing but your glycerine. That one. Okay. And this right here, students, this right here is called as glycerol also. Glycerol. Okay. Glycerol or glycerine. Colorless, sweet and viscous in nature. Fatty acid plus glycerol. It's a type of alcohol. My dear friend, ulcer. Ulcer is your friend. Oh my god, ulcer is very bad. One suffocation it is. Once you get the ulcer, right, you can't even eat anything. You don't want to eat food only. The sambar to forget. If sambar goes in the ulcer, you feel like removing your mouth and throwing your mouth only somewhere. Okay. So this is called as glycerol. Now students, tell me, can we take this fatty acid and this glycerol, can we join them? Tell me, can we take this glycerol, fatty acid and glycerol and completely join them into one bundle now? Yes. Can we join them? Tell me. Yes, we can. If you want to join them, the process is called as esterification. The process is called as esterification is the process when you try to join them. If you want to join them. So let's join them now. First of all, tell me, let's do glycerol. Let's do glycerol. OH, 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 H2, H2. Okay, this is one here, 2 hydrogen. This is glycerol. Let's take one simple fatty acid. I'll tell you the examples later on. Let's take one fatty acid. Let's take palmitic acid. Palmitic acid is 16, right? CH3, CH2, 14, 15, 14, no, 14, yes, 14, 14, 15, and there should be one COH also. COH, right? Students, remember this entire thing. This is 14, okay? 14. This entire thing right here is the R. This entire thing is the R group. This is the carboxylic group. It's a fatty acid. This is a fatty acid. This right here is a glycerol. This is a glycerol. Here, fatty acid. Did we learn here? Fatty acids have one carboxylic group and one R group. So R and carboxylic group. Same thing we are writing. Same thing we are writing here. Okay. Now let's do bond formation. We can take this particular hydrogen and this OH here, and baby will happen between them. What do we obtain, students? What do you obtain now? Okay. O, C double bond O. Okay. So what do we get now? We have the carbon. We have H2. OH. We have only O here. Only O. Then C. This is double bond O. Right. Then we have the CH3, CH2, 14. Then we have the CH2 14 times and CH3 and CH3. Yes. Students, when glycerol plus fatty acid, we end up getting a baby. The name of the baby is called as glyceride. It's called as glyceride. Now students, did we just replace only one of the OH? Only one of the OH? Yes. So can I call this as monoglyceride now? Monoglyceride. Only one OH was replaced. Okay, water is also coming out right here, minus H2O. So can I call this monoglyceride? Yes, monoglyceride. Now if I add one more fatty acid here, students, if I add one more fatty acid here, then I'll call it diglyceride. If I add one more fatty acid here, then can I call it a triglyceride? Are you getting? So monoglyceride only one, the hydroxyl group is replaced. 2 diglyceride. 3 is called as triglyceride. That's all. Oof, such a chemistry, no? I don't know how. Chemistry is fun to teach also. Very simple is chemistry, but so the structure is important to learn. No, they will never ask you the structure. But I want you to know, they'll never ask you the structure. This is for concept building. You need to understand what is the meaning of glyceride. Because the NCERT is mentioning this. One line is mentioned in NCERT, but I'm explaining that one line with the help of one slide here. I'll show you exactly where it is. Okay, now students, can we read lipids now? Can we read lipids? Can we read lipids? Here, what is given in lipids? Understand. Lipids are generally water insoluble. Obviously, can you mix oil and water? You can mix, but usually oil will float on water insoluble. They could be simple fatty acids. A fatty acid has a carboxylic group attached to an R group. Did we learn that? Did we learn that, students? Fatty acid is carboxylic plus R group. Okay. The R group could be a methyl, ethyl, or any number between 1 to 19. For example is very important. Write down. Example is palmitic acid. First example is palmitic acid. Palmitic acid is how many carbon? 16 carbon. Okay. Then arachidonic acid. Then we have arachidonic acid, which has around 20 carbon, including the carboxyl carbon. See, it always includes carboxyl. That's why when I wrote here, students, when I wrote in esterification, I wrote here 1, 14, 15 and 16. So 16 carbon here. So it's really fun to teach. I took seminar. See, teaching is always really fun. And if you really enjoy teaching, it's the best job in the world. For me at least. Okay. Now, fatty acids could be saturated. That is, without double bond means single bond. Right. Or unsaturated with double bond. Did we do that here? Yes, we did that. It can be saturated or unsaturated, right? Now, another simple lipid is glycerol, which is trihydroxypropane. Trihydroxyl or trihydroxypropane? Trihydroxypropane. Okay, trihydroxypropane. So what is palmitic acid? Palmitic acid is acid, students. It is a type of your acid, which is a type of your fatty acid. Type of fatty acid with 16 carbon. Long chain of 16 carbon fatty acid is there. Okay. Okay. Now, many lipids have both glycerol and fatty acid. See, many lipids have both. I told you, they should make a baby here. Here, fatty acids are found esterified with glycerol. See, this is a simple AR question, assertion reason question. They'll ask you, assertion is going to be glycerol and fatty acids can be esterified. Assertion. Reason, they will result in a glyceride. Do you realize they can ask such a question in examination from one line of NCERT? Unless you know the concept of esterification, which I just taught you, this line would have been difficult to understand. Here. Okay. They can be then monoglyceride, diglyceride and triglycerides. These are also called as fats and oils based on the melting points. Oils have lower melting point, gingerly oil. See here, gingerly oil. And hence remain oil as winter. Okay. Some lipids have phosphorus. They are called as compound lipids. They are basically compound lipids. Okay. Example very important. Phosphorus and phosphorylated organic compounds in them. These are phospholipids. Very important. PYQ. Example of phospholipid is going to be lecithin. Lecithin is an example of phospholipid. Okay. They are found in cell membrane. Where do you find lecithin, students? Lecithin is found in the cell membrane. Okay. Cell membrane. Gingerly oil. Can't get over it. Lecithin is one example. Some tissues, especially in the neural tissue, that is your cerebroside and everything. Some tissues, especially in the neural tissue, can have lipids with more complex structure. That is, lipid present in your brain. Lipid present in your white matter. White matter also has lipids. Okay. Lipid is good. Gingerly oil will get hurt. Huh? Who keeps the name gingerly oil and not expect to laugh about it? I don't understand. It sounds funny, so funny. Students, with that your lipids is also over now. Lipids is also done and dusted. Done and dusted. Okay. Now students, next topic is going to be, can you hear me right? You can hear me right? It is done. Students, tell me, is it easy, right? Tell me, is this chapter difficult? Is this NCERT even difficult to understand for this chapter? Very easy it is. So similarly now we will go with something called as nucleic acid. Right, we will go with nucleic acid. How much is there after today's class? Nucleic acid is there. What is DNA, RNA? I'll tell you. What is DNA? What is RNA? I'll tell you. What is pyramids, pyramids and purines? All of you know that. Don't act smart now. I know all of you know that. Pyramids and purines. Okay. Now, then I will teach you something called as secondary metabolite, which will take around 15 minutes of time. 15 minutes secondary metabolites, not 15 minutes. 10 minutes secondary metabolites. Then we will do something called as biomacromolecules, which all of you know already. All of you have know this. I have already explained this to you. Okay, biomacromolecules, and it will be over later on today's class. Then we will do questions. Now students, 1.5 hour, 1 hour 45 minutes. My legs are a little shaky now, so I will go sit for like 15 minutes and come back. What's that? Okay, I will go for 15 minutes. I will sit aram se for some time, drink some water. Okay. And think about my lunch, not eat my lunch obviously. If I eat lunch and come, I will start yawning in the class, right? So I can't eat lunch and come. I will eat the lunch after the class, but I will go for sit down for 20 minutes, something. I'll take my rest little. Then we will finish what is nucleotide, what is secondary metabolite, and some PYQs. Okay, then we'll do some PYQs. So what is purine? Nice. I don't know what is purine. So purine is a biscuit name. Biscuit it is, purine. I'll give you the biscuit later on. Wait. Okay. Purine is a biscuit. Pyrimidine is a special type of biscuit. Purine is a biscuit. Okay, I'll tell you the biscuit later on. So I will sit for 15 minutes and come. And 20 minutes probably I'll go eat some biscuits. Actual biscuits, not purines. I'll go eat actual biscuits. I'll come. Okay. So all of you sit tight and come back in next 20 minutes. Then we will do full-fledged high Josh. We will do the remaining part. Don't run away anywhere. Okay, don't run away anywhere. Be here in the class only. Because when I come back, I know all of you will run away somewhere. Okay. So what is the time right now? It's 2:51. So we will start the class at 4:10. 4:10. What's it? 4:10. Okay. 4:10 class resumes. Resume at what's it? Okay. So sit at everyone. Go eat something. All of you go eat something. Don't eat very heavy, otherwise you will sleep in my class. Okay. So go eat something, drink some water, and chill for some time for 20 minutes. Then come back with all the brain cells activated, and we'll kill it today again. Okay. I'll come back.
So hello students, welcome back. Yes. All of you, welcome back to the class now. Welcome back to the class. Okay. So let me know in the chat if I'm audible and visible, all of you quickly in the chat. Why there are so many deleted messages? Okay, it's fine. Let's see what happens. So let's start with the next topic right now. That is going to be your nucleotides. The next topic for right now is going to be nucleotides. Let's read what is given in NCERT. Let's understand what is given in NCERT about your nucleic acids now. Nucleic acids, right? Nucleic acid. First of all, tell me students, nucleic acid are a part of your macromolecule or a micromolecule. Nucleic acid. Nucleic acids. So all of you remember students, we learned here also. I keep going back unless you remember this throughout. If you come back here, did I mention nucleic acid are a part of your macromolecule? Nucleic acid are a part of your macromolecules, yes or no? Yes, they're part of macromolecules. So they're part of macromolecules. Macromolecules. So can I tell their size is more than 1000 Daltons? They are found in acid insoluble fraction. Yes. The nucleic acids, since they are found in acid insoluble fraction, can also they're found in retentate. Yes, they're found in the retentate. Nucleic acid. Now students, when many nucleic acids join together, they make the nucleotide. They make up the nucleotide. Okay, the polymers of nucleotide. Nucleic acids are nothing but polymers of nucleotide. When many nucleotides join together, we get the nucleic acid. Living organisms have a number of carbon compounds in which heterocyclic rings can be found. Heterocyclic rings. Okay. Some of these are nitrogenous bases: adenine, guanine, cytosine, and uracil and thymine. All 5 of them are very important. When found attached to the sugar, they are called as nucleoside. So what is a nucleoside, students? Nucleoside is nothing but hydrogen base with a sugar. What is a nucleoside? Nucleoside is nothing but a hydrogen base plus a sugar. So what is a nucleoside? Nucleoside is nitrogenous base plus sugar is a nucleoside. Then what is a nucleotide? Nucleotide is going to be plus phosphate. Phosphate plus sugar plus the nitrogenous base. All 3 together is a nucleotide. While your nucleoside is nothing but a nitrogenous base plus a sugar, that is a nucleoside. So learn the difference between a nucleotide and a nucleoside. Okay, so nucleotide plus nucleoside. Okay, so what will be? If a phosphate group is also found, esterified to the sugar, they are called as nucleotide. Adenosine, see, sin will always represent a nucleotide. Adenosine, guanosine, thymidine, uridine, and cytidine these are nucleosides. They are nucleosides. Tide is going to be acid. Adenic acid, thymidinic acid, guanic acid, uric acid, cytidinic acid are nucleotides. So acid is a nucleotide. Okay. Nucleic acids like DNA and RNA. They are nucleic acids like DNA and RNA consist of nucleotide only. They have nucleotide, right? So DNA and RNA will have a nucleotide, right? So they will have a phosphate attachment, sugar attachment, and nitrogenous base attachment. Okay. Now, DNA and RNA function as genetic material. All of you know DNA and RNA function as genetic material. So here is your nucleotide. Do we have phosphate? We have the pentose sugar, phosphate, and nitrogenous base. All 3 components together is called as a nucleotide. Now, what is the difference between DNA and RNA? DNA is called as
DNA is called as deoxyribonucleic acid. DNA is called as deoxyribonucleic acid. RNA is called as ribonucleic acid, RNA. The major difference lies in the structure of sugar. Structure of sugar of DNA and RNA. If you look at the second carbon, right here, if you look at the second carbon of RNA and the sugar present in DNA, you will notice the meaning of D. D means something is missing. Deoxy means oxygen is missing in the second carbon. Can you see here? There is no oxygen is here. Ribonucleic acid. Ribonucleic acid has the oxygen here next to the hydrogen in the case of second carbon. That is the structural difference between a DNA and RNA. Deoxy, D means missing, oxygen is missing in the case of second carbon of your deoxyribose in DNA. Ribose sugar has the RNA. In RNA they have the ribose sugar has the oxygen is present. Difference between D and RNA structurally. Now students, then we have purines and pyrimidines. Remember this, for purines, my trick is very simple. Air is pure. There are many tricks in the world, but for purines it is air is pure. A is nothing but adenine. Pure. Air is pure, right? And P is made, air is basically made up of many gases. So guanine. And P is also for purines. But I'll tell you an easier way to remember this. Can I tell you an easier way to remember this? Very easy way to remember this. Learn pyrimidines, you can remember purines easily. If you learn pyrimidines. How do you remember pyrimidines? It's very easy to remember pyrimidines. Pyrimidines can be written as cut pyramid. Just learn cut pyramid. And you can remember. That's all you have to remember. Cut pyramid. What is cut? Cut is nothing but your cytosine, uracil and thymidine. Cytosine, uracil and thymidine are your pyrimidines. Learn pyrimidines. Whatever remaining two are going to be purines. That is adenine and guanine. Okay. Cut pyrimidines. Now they'll ask you a question. There's one famous question here. They'll give you four options. They'll give you options such as cytosine, thymine and uracil. Option number 1, option number 2, option number 3 and option number 4 is adenine. And they will tell you to find which of the following is a double benzene ring structure. Which of the following is a double ring structure? What is your answer? Tell me then. Double ring structure. Out of cytosine, thymine and uracil and adenine, which of the following is double ring structure? What is the answer then? Students who are coming late, quickly smash the like button, mark your attendance even if you're watching it recorded. What is the answer? Double ring structure. See students, if you look at the structure of cytosine, thymine and uracil, single ring. Look at the structure of adenine and guanine. Ring 1, ring 2. Double ring structures. They are double ring structures, that is purines are double ring. So obviously then the answer would be option number 4. Option number 4 would be the answer. Okay, adenine. Now students, I've already told this. All of you know this. All of you know ATGC, right? All of you know ATGC rule. ATGC. Now I'll ask you a question. In a DNA, question is very simple. In a DNA, there is 30% adenine, 20% cytosine. What is the percentage of thymine and guanine? Thymine and guanine. Tell me. What is the percentage of thymine and guanine? Simple question. In a DNA, 30% is adenine, 20% is cytosine. What is the percentage of thymine and what is the percentage of guanine? Friends, all of you know if adenine and thymine is going to remain the same. So thymine is going to be again 30% and guanine is going to be 20%. That's all. That's all is the answer. That's all is the answer. So they can ask you such questions to confuse you. But it is going to be very easy answer. It is going to be very easy answer if you know the ATGC rule. ATGC rule if you know it. Okay. It's a very simple question. That's why. Now, all of you know this? Let's read what is given in NCERT. Did we read that? Did we read this? Yes, we read that. All of you know phosphate, sugar and base, nucleotide. Sugar and base is called as nucleoside. Phosphate is missing in nucleoside. See, this is the structure of palmitic acid here. They're given the structure of palmitic acid here. Palmitic acid. 16 carbon. Glycerol. 1, 2, 3. 3 OH. 3 carbon. 5 hydrogen. Triglyceride. Every single hydrogen has been replaced. Triglyceride. Cholesterol is a type of lipid. Then we have lecithin is a phospholipid. Present where? Present in the plasma membrane. Present in the plasma membrane. All these structures are very important to understand. Cholesterol. Cholesterol is a derived lipid. Derived lipid. Okay. Did we learn this? Adenine, double ring structure. Adenine, guanine, double ring structure. Right? Double ring structure. Adenosine, uridine are nucleosides. Nucleosides. Okay. Nucleoside. If there is a adenic acid, it is called as nucleotide. 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 Okay. Done. So with that, your nucleic acid topic also is over. In just 10 minutes. Just 10 minutes, topic is over. We are covering every level of NCERT. It is so easy, right, to understand once you listen to the content, right? The next thing is going to be something called as primary and secondary metabolites. I'll read it for you. I'll tell you what exactly is important. That's all. Okay. The most exciting aspect of chemistry deals with the isolation of thousands of compounds, small and big, from living organisms, determining their structure and if possibly, synthesizing them. In chemistry lab they do. Right, you take one compound, you create them, recreate them. Okay. If one way to make a list of biomolecules, such as a list would have thousands of organic compounds, including amino acids, sugars, etc., for a reason that is given in the section 9.10, we can recall these biomolecules as metabolites. Right. Some of the biomolecules can be called as metabolites. In animals, one notices the presence of all such categories of compounds shown in 9.1. Basically, the proteins are present in the body, carbohydrates are present in our body, lipids are present in our body, right? Amino acids are present in our body. So all of these metabolites are present in our body and it is required for our body. Now tell me, Tanuja, from Tanuja's body, if you remove all the protein, will she survive? If you remove from the Ravindranath's body, if you remove all the lipid, survive? No. If we removed from Rishita all the amino acids, will the person survive? No. So these metabolites are very important for the body. Without these metabolites, primary metabolites, you cannot survive. You cannot survive. These are called as primary metabolites. However, one analysis of plant, fungal and microbial cells, one would see thousands of compound other than the primary metabolites, such as your alkaloids, flavonoids, rubber, essential oils, antibiotics, colored pigments, scents, gum, spices. All of these are called as your secondary metabolite. Secondary metabolites. Secondary metabolites are not essentially for survival, but they are for the defense mechanism or something. Okay, see here. While primary metabolites have identifiable function to play to role in the normal physical process, we do not at the moment, that's defense mechanism only, we do not know at the moment understand the role of functions of all the secondary metabolites. Some functions are known, okay, in the host organisms. However, many of them are useful to humans. Example, rubber. Yes, we make pen, this rubber tip. Yes. Drugs. Yes, not the drugs which you're thinking of, not that drugs. Drugs here means medicine. Drugs here means medicine. Okay, means medicine. Okay, not other drugs and all. Don't worry about that. Spices. In biryani, if you don't add the spices, will it be tasty? No. Right. Scents, the perfume which you add. But now it's mostly chemical, but earlier it was natural. Right. And pigments, the color pigments you add, no, in the different things, pigments. Some secondary metabolites have ecological importance also. Okay. In later chapter you will learn that. Here is a list. Remember the list. We will play a small game now. I will ask you a question and you have to tell me the answer. You will have 1 minute to read the list and remember it. Okay. Read the list. I will play a small game. I will ask you a question from this list and let's see how many of you remember. Time starts now. Go. Time starts now. Pigment, remember, carotenoids, anthocyanin is a pigment. Alkaloids, morphine and codeine. Morphine, you know, right? It is used as a drug, right? Codeine, cough syrup will be there, right? Terpenoids, monoterpenes and diterpenes. Essential oils, lemongrass oil. Essential oil, they are, no, the candle they have, the scented candles. Even I have one in the house. Nice they are. Okay. Toxin is arabrin and ricin is a toxin. How do you remember it? Iron. Toxin, arabin and ricin. Lecithin is concalvaline. Okay, it's a type of lecithin. It is a type of your, it initiates root initiation also does. Drugs is vinbrastin, which is present in your vinca rosa, which helps in cancer treatment. Curcumin, from your curcumin longa. Curcumin. Okay. Not cucumber. Curcumin is your turmeric. Okay, not cucumber. Polymeric substances like your rubber, gums and cellulose. Okay. Read this and I'll ask you the examples. Tell me once you're done, all of you. It's a small game. I'll give you 1 more minute. Read all of them. Remember. Then I'll ask you a question. Okay. Done. Oh, okay. The question is, concalvaline A is which type of secondary metabolite? Concalvaline A is a which type of secondary metabolite? Drug, alkaloid, right? Which type of secondary metabolite is it? It's a pigment. Concalvaline A, all of you. Answer in the chat. Concalvaline A. I'll give you 10 seconds. 10, 9, 8, 7, 6, 5, 4, 3, 2, 1 and time's up. Concalvaline A is a type of lecithin. Concalvaline A is a type of lecithin. Okay. One more question now. Okay. One more question. Codeine is a which type of secondary metabolite? Codeine. Go. Codeine is a which type of secondary metabolite? Codeine. 10, 9, 8, 7, 6, 5, 4, 3, 2, 1. Let's see who answers first now. Codeine, answer. I want to see. Codeine is actually a type of alkaloid. Codeine is a alkaloid, not drug. Abby, exactly. It is a type of alkaloid. Exactly. Okay. Can we go for one more? Now? One more. Lectin. Not lecithin, no? Lectin. Lectins. Next one more, huh? Okay. Curcumin is which type of secondary metabolite? Curcumin. Curcumin, go. Curcumin is a which type of secondary metabolite? Curcumin. Curcumin, go. Curcumin, curcumin longa. 10, 9, 8, 7, 6, 5, 4, 3, 2, 1. Absolutely. Curcumin is a type of drug. Students, this is a very important table. I cannot stress enough because many questions have come from here. Okay. Now students, can we start the last part of today's class now? After this I have some PYQs. We will solve some PYQs and you can go tata bye bye, enjoy your life. Okay. Can we start? Last part. I'll read it. You already know all of this. So let me read it for you. Okay. There is one feature common to all the compounds found in the acid soluble pool. Down. Okay. They have a molecular weight ranging from 8 to around 800 Daltons. That is less than 1000. Also. Acid insoluble pool on the top has only 4 types of organic compounds. That is proteins, nucleic acids, polysaccharides and lipids. Nucleic acid, which we already done. Proteins and carbohydrates in the next class. Lipid also is done. Right. These classes of compounds, with exception of lipid, obviously lipid is less than 1000 Daltons, have molecular weight in the range of 10,000 Daltons. Very high. Okay, or above. For this very reason, biomolecules, that is chemical composition found in living organisms, are 2 types. One, those which have molecular weight less than 1000 are usually referred to as micromolecules or simply biomolecules, while those which are found with acid insoluble fraction on the top is called as macromolecules or biomacromolecules. Okay. The molecules in insoluble fraction on the top, with exception of lipid, are polymeric substance. Means it can make polymers, long chain polymers it can make. Nucleotide can join together and make nucleic acid. Right. Amino acids can join together, can make a protein. Right. Carbohydrates, long chain of carbon can make carbon and hydrogen can make a carbohydrate. Polysaccharides, many sugars can join and make a polysaccharide. They are polymeric in nature, but lipid is not polymeric in nature. Then why do lipids of whose molecular weight is less than 1000 Daltons, do not exceed 800 Daltons, come under acid insoluble fraction? That is, lipids are indeed small molecular weight compounds, but are present not only as such but also arranged into structures like cell membrane and other membrane. When we grind a tissue, we are disrupting the cell structure. Cell membrane and other membrane are also broken into pieces and form vesicle, which are not water soluble lipid vesicles. Therefore, these membrane fragments in the form of vesicle gets separated along with the acid insoluble fraction. That is, lipid forms a vesicle and it is present on the top, acid insoluble fraction. Hence, in the macromolecule fraction, lipids are not strictly macromolecules. See, very important line. Lipids are not strictly macromolecules means what? It means that even though their molecular weight is less, they are in the acid insoluble fraction. So they are macromolecules, but not very strictly. With the size, yes, with the size they will be coming in micro. But since they are forming vesicles, they are on the top. Acid soluble fraction represents roughly the cytoplasmic composition, down part. The macromolecules form the cytoplasm and the organelles become the acid insoluble. So cytoplasmic composition is the cytoplasm is present on the bottom. Rest of the part is on the top. That is, cytoplasm and mainly the organelles are present on the top, acid insoluble fraction. Together they represent the entire chemical composition of living tissue or organism. In summary, if we represent the chemical composition of living tissue from the abundance point of view, arrange them in class wise, we observe that the water is the most abundant chemical in the living organism. Very important line. Water is the most abundant chemical in the living organism. See, water. 70 to 80% of the cell is made up of water. Okay, made up of water. Then is proteins, then carbohydrates, then lipids, then nucleic acids, and then only 1% is the ions. Okay. Here is the question. Read the question and tell me the answer. Amino acids have a property of ionizable nature, NH2 and COH groups, hence they have different structures, different pH. Yes, they have different structure, different pH. Amino acids can exist as bitter ion form at acidic and basic pH. No, they are not found in acidic and basic pH. Aren't they found at a special pH? The special pH is called as what? Isoelectric pH, and isoelectric pH is almost a neutral pH. It is almost a neutral pH. Right? It is almost a neutral pH. So can I tell this statement is wrong? Yes. This statement is wrong. Actually, see, there are a lot of confusion regarding the second statement, students. There is a lot of confusion regarding the second statement. It says that amino acids can exist as bitter ion at acidic and basic pH. Students, do you remember I told you? In acidic pH, they will be protonated. I'll tell you here. Let's clear this confusion today only. Many places they have given this answer also wrong. Okay, let me show you. In acidic pH, will be acidic pH. This is how you get. In basic pH, this is you get. But at a particular pH called as isoelectric pH, at a particular pH called as isoelectric pH, which is almost neutral pH, you get the zwitter ion. So zwitter ion is only available at a neutral pH called as isoelectric pH. Remember that, all of you. Okay. Oh, very less questions are today. Less questions are there. Okay. Easy question. Oh, no, this should be the answer. This is not. This should be the answer. Statement 1 is correct. Statement 2 is incorrect. This is the answer. Okay. Not the second. 3 is the answer. This question. Identify the basic amino acids from the following. Very easy question. Laddu question. This is. Start. Laddu question. Identify the basic amino acid from the following. Identify the basic amino acid from the following. Done. Apart from lysine, can anyone tell me what are the other basic amino acids? Lysine, arginine, and histidine. 3 are very important. Tell me, what about glutamic acid? Glutamic acid is an acidic amino acid. It is a acidic amino acid. Valine is a neutral amino acid. Tyrosine is an aromatic amino acid. Okay, it's an aromatic amino acid. Now, the following can be called as zwitter ion. Which of the following can make zwitter ion? Tell me. Which of the following can make a zwitter ion? Tell me. Fatty acids cannot make it. Monosaccharides, no. No. Only an amino acid can make a zwitter ion. Read the following statements on lipids and find out the correct set of statements. Okay. Lecithin is found in plasma membrane. Is a glycolipid? No. Lecithin is a phospholipid. Okay. Saturated fatty acids possess one or more double bond? No. They will only have single bond. Unsaturated has double bond. Okay. Gingerly oil has low melting point. Yes. Low melting point and remained oil as winter. Yes. Gingerly oil, low melting point. Ghee, high melting point. Correct. Lipids are generally insoluble in water but soluble in some organic solvents. Yes. When fatty acid is esterified with glycerol, we get monoglyceride. Yes. So CDE. CDE, right? Option number 4. Option number 4. CDE. Lecithin is a phospholipid, not a glycolipid. Next question. Lipids have only single bond are called as unsaturated fatty acids. No, students, no. Single bond means what? Saturated. Lecithin is a phospholipid. Yes, it is a phospholipid. Yes. Trihydroxypropane is called as glycerol. Yes. Palmitic acid is 20 carbon. No. Palmitic acid is 16 carbon. Arachidonic acid is 20 carbon. So what is the right? Right is B and C. B and C is option number 1. Option number 1. A for AIMS. A for amazing copy, ma'am. Okay. Which of the following is not a secondary metabolite? See, curcumin, yes. Morphine, yes. Anthocyanin is a pigment, yes. But lecithin is a type of your phospholipid, not a secondary metabolite. Imposter is there. So option number 4. Diksha ma'am. D for Diksha ma'am. Yes. Diksha ma'am. Next question. Match the following. Adenine. Adenine is a purine. A is 4. A is 4. See, these two are gone. A is 4. Anthocyanin is a pigment. B is 1. C is 2. Chitin is a phospholipid. Yes, polysaccharide. Codeine is an alkaloid. We just now learned today. Codeine is an alkaloid. Yes, number 1. Which of the following is not a secondary metabolite again? Not a secondary metabolite. I'll give you 10 seconds, go. I'll give you 10 seconds. Not a secondary metabolite. When blasting curcumin, rubber, gum, morphine, codeine, amino acids and glucose. Amino acids and glucose. Exactly, Ravindrani. The 4th one. Amino acid and glucose are not of a type of your secondary metabolite. Secondary metabolite. This is your homework question. It's a NEET 2024 question. Homework question. Take it down. Let me know in the comment section. I will check today. Yesterday I checked, many were not there. So I thought I'll not give it only this year, this time, next class, but I'm giving it next class. So here is the homework question. So students, the part 1 made you realize that there is something called as, what we learned today? We started off with something called as experiments, right? Both experiment. Something called as a normal chutney making, otherwise ash test. Then I told you something called as, we learned about your amino acids. We learned the structure of amino acids, 3 different structures, basic amino acid, acidic acid amino acid. We also learned about neutral amino acids, essential, non-essential amino acids, aromatic amino acids also we learned. Then we went on to learning about lipids and how are lipids. In lipids we learned about fatty acids and glycerol. And if you fuse them, acidification glyceride. Then we also learned about primary, secondary metabolites. We learned about nucleic acids, right? We learned about nucleic acids, DNA, RNA, and we also learned about what type phospholipid. Then we learned about something called as what is that? We learned there nucleotide, nucleoside, right? Then we learned about primary and secondary metabolites. Then we had a general overview of biomacromolecules. Biomacromolecules. So that's the entire of today's class. And let me tell you students, the next class is going to be even more important from today's class. Why? In the next class, because we will be covering more high weightage chapters, topics. In the biomolecules part 2, I will be teaching you something called as the enzyme reaction. Competitive, uncompetitive, non-competitive. I'll be teaching something called as enzymatic reaction, right? And I'll be teaching something called as structure of protein. Primary structure, secondary structure, tertiary structure, quaternary structure of protein. Then I'll also be teaching something called as carbohydrates, monosaccharide, disaccharide, oligosaccharide, polysaccharides. Functions of polysaccharides. All of that is very important, and let me tell you, 60% of questions will come from that side, part 2. 40% will come from today's class. So if you want the 100% completion in your biomolecules, you need to watch both the lectures. So let me know in the comment section if you're excited for the part 2, which is going to be amazing. We will be talking about NCERT line by line, solving PYQs, detailed fashion. Any doubts I'll be answering, and I will see you in the next class. And students, this is a quick reminder for all the students to write the MVSAT exam. because it's right around the corner. And all of you know the advantage of MVSAT. Link in the description. Quickly go find offline or online which is suitable for you. Okay. So all of these notes will be available. Next part will be in the next week. Next week, probably let's say Tuesday. Let's do it on coming Tuesday, right? Tuesday. Let's not keep a lot of gap. So Tuesday, what say? So this coming Tuesday, we will do the part 2. Sounds okay for you? So Tuesday, so all of you will be aware of it. And you can go and mark your attendance also. Tuesday. The coming Tuesday, 6 p.m. part 2. The coming Tuesday. Okay. Coming Tuesday, we will do it. So all of you can remember it also. You can join the class. You can learn it also. Okay. So after biomolecules, what do you want, students? After biomolecules, I was thinking, should we do line by line of morphology in flowering plants? See, I know morphology you have learned. But we have not done line by line of it, no? We have not done line by line of it. So I was thinking we will do morphology of line by line, and we will complete, or we will do respiration in the plants. Your call. Or we will do respiration in plants. Respiration in plants is a very easy chapter. So let's do respiration in plants. Any one of those we will do. Respiration, morphology or anatomy. Very easy, all of them are. Okay. Let me know in the comment section what you want in the next class also, after in NCERT line by line. Okay. Don't tell physics now. Physics and chemistry. You will fail if you do physics and chemistry from me. Okay. So, bye-bye. Take care, all of you. Have a great day ahead. Happy Ganesh Chaturthi, all of you once again. Happy Ganesh Chaturthi. So, be serious. I'm telling you again and again. I'm reminding all of you again and again, okay? I'm reminding again and again to all of you, be serious. 239 days are left. And by the time you're watching this video, if you're already late, even more less time. So, students, show your motivation. Keep your motivation high. Be motivated. Yes, you can do it. And believe in us. I believe in all of us. We are doing all of this. See, students, yesterday night we took a class here. All of you were sitting with me at 9 o'clock till 9 o'clock. You were sitting with me yesterday night. Again, next day itself, you're doing biomolecules with me. So that shows the motivation which you have. And hopefully that is reignited and you'll have the same josh throughout your preparation now. So keep the motivation high. Study well. Okay. So bye-bye all of you. Photosynthesis chapter notes I will send today only. Don't worry. It will not be delayed. Okay. So thank you so much, students. Bye-bye. Boy. Take care, all of you. Homework question in the comment section. Do not forget. I will check.